Welcome back to summary video for the Water for Transport chapter. This will cover videos 7 to 12 of the Water for Transport playlist. I'll start with the first one. That was describe the main changes in the chemical composition of blood as it moves around the body and identify tissues in which these changes occur. So here we have to actually talk about what changes in terms of chemicals that go in or out of the blood when it travels from the heart to the other parts of the body. So I'm going to go through each as well, step by step. The first one was when it reaches all these pink areas, the muscles, the brain or the tissue, the muscle or the brain tissue, so muscle like the leg or the arm muscle tissue. So what happens to the blood? So we have three things happening. We've got glucose and oxygen going in, so they're going in because of cellular respiration, and then we also have through cellular respiration we create CO2 as a waste product, and that will go into the blood. So what goes out of the blood is glucose and oxygen, and the reason why is because we need to have that for cellular respiration at the cells. These are all cells. And what goes into the blood is CO2. CO2 is that waste product that the cells are producing, the garbage that the cells are producing that they're going to dump into the blood. So that happens at the muscle and brain tissue. At the small intestine, we're going to have glucose and amino acids. So if we have eaten food, it's going to be full of glucose and amino acids, or proteins and carbohydrates. They're going to be broken down into glucose and amino acids. And at the small intestine, that's where we're going to absorb them from our small intestine into the blood itself. So all our food particles, broken down food particles, are going to be absorbed into a small intestine. We're also going to absorb vitamins and salts and everything else also from the small intestine here. Next will be the lungs. So at the lungs we have oxygen going up and out in terms of our blood and CO2 going down. The reason why is you have these blue sacs. Remember there were the alveoli. And the alveoli you have oxygen diffusing over. So we have been breathing in oxygen, and that diffuses over into the bloodstream, which means our oxygen in our blood will go up. And CO2, which was the waste product of cellular respiration, comes all the way to the alveoli, to the lungs, and that's where we exhale it. That's where we remove it, and it goes from the alveoli, and then from the alveoli out through our nose or through our mouth. And so that was at the lungs. We have oxygen increasing and CO2 decreasing in terms of the blood. Then we are at our liver. In our liver we produce urea, so urea is produced here. Liver produces urea, which means that our liver cells will actually dump urea from their cells into the bloodstream. So what happens is urea increases in our blood at the liver. And then we travel all the way to a kidney. If you haven't done the third chapter yet, the kidneys are responsible for urea removal, and this is supposed to be the nephron. So that's the cell of the kidney. And you have urea, which is now in our bloodstream, going from our bloodstream into the kidney. So our blood loses urea because it's been removed by the kidney, which is good because urea is a waste product as well. Right, so those were the main changes. So remember that the areas that it happens and what happens as well. So then we have this one as well, which is outline the need for oxygen in living cells and explain why the removal of carbon dioxide from cells is essential. So remember why we needed to have oxygen. Oxygen was important for cellular respiration. So this was the equation of cellular respiration. And cellular respiration was really important because it allowed us to produce energy. And remember, energy was really important for things like growing or moving or reproducing or repairing our body. So without energy, we wouldn't be able to do anything. And without oxygen, we wouldn't be able to make energy. So we need to have oxygen for cellular respiration to make energy, and energy allows us to survive. Also, we need to remove CO2, we need to remove carbon dioxide. The reason why, if you remember, if carbon dioxide reacts with water, and that water can be either in cells or in the plasma itself, what happens is we produce these hydrogen ions, and what they do is they make our blood or our cells more acidic. And remember that optimum pH, so that was right here. If it goes too low, which happens if we have too much CO2 in our body, it will decrease, our pH will decrease, which means our enzymes, this is when it's normal, this is when it's optimum. But when it's decreased, or when it's increased, but CO2 decreases our pH, what that does, it denatures our enzymes. So it makes that not work properly anymore. And if our enzymes denatures, that means our metabolic activity has gone down, and that means we will eventually suffer a lot from that. So we need to make sure our enzymes are normal, and we do that by removing the CO2 that we produce. Components of this, this one is identify the products extracted from donated blood and discuss the uses of these products. So we have to name them and we have to discuss the uses. So remember, like if you were to cut yourself, what you would see is you see that red blood. That's called whole blood, because that's basically everything that is in our blood all at once, because we haven't filtered anything out yet. And people donate whole blood, and whole blood is often used for accidents, rapid blood loss, or anemia. So if you have an accident or if you have anything that you lose lots of blood, you need to have every single component that is in the blood to replace that. Anemia was low iron levels. And remember, iron was in red blood cells because hemoglobin, so heme was for the iron. So if you are given whole blood, you'll also be given red blood cells, which means you're going to increase your iron levels. Low iron levels. But we can also break the actual red blood cells or the whole blood into its components. It's like taking apart a car. If the car is whole, it's called whole blood. And if the car is broken into its parts, we call those the components of the blood, or the components of the car. So the components of the blood are these. We've got platelets, red blood cells, immunoglobulins, white blood cells, and plasma. And I'll describe the uses of these products to identify the components and the uses. Platelets, they're used for platelet deficiency disease. That's when people have not enough platelets for whatever reason. And leukemia. Leukemia is a form of cancer, often present in children. Not often, but it's most common in children. And leukemia, people will have low platelet counts. So by injecting only platelets, we make sure that their platelets numbers have increased. Red blood cells, again, that was used for anemia. That was low iron count. So if someone has low iron levels, we give them red blood cells. The reason why is because red blood cells has hemoglobin, and that heme stands for iron. If people have low iron levels, that means they have low oxygen levels as well. By giving them red blood cells, we increase their oxygen carrying capacity, how much oxygen they can carry in their blood, because of that hemoglobin, which is in red blood cells. Immunoglobulins, these were antibodies. So immunoglobulins were antibodies, and antibodies help fight infection or disease, especially for some of the ones which are hard to treat, so measles and tetanus. So one person might have an immune antibody or antibodies against these two different diseases. So they get the immunoglobulins from that person and inject it into a person who has that disease but who doesn't have the antibodies. So immunoglobulins help fight disease such as measles and tetanus. So they're used in treatment. White blood cells, they help fight infection in general. They're quite general in terms of what they do. And you give it to people who do chemotherapy, because people who do chemotherapy, usually cancer patients, this is often involved cancer. We give them chemotherapy to kill their cancer cells, but the problem is we also kill their good cells as well, so many of the white blood cells will also die. So for people who lose all the white blood cells because of cancer, we inject other people's white blood cells into their body to make sure that they can still keep fighting infection whilst they undergo chemotherapy to kill their cancer cells. And then we've got plasma, and plasma was there for coagulation problems, so people who have too thin blood, we give them plasma. The next one was use available evidence to propose reasons why artificial blood is needed, and how far we are with making artificial blood. So how far are we with making artificial blood, and why is it needed? I'll start with why it's needed. Longer storage, so it lasts for more than a year, compared to 1 month or so for normal blood. We can make more of it, so at the moment we have a huge shortage of blood. People don't donate blood, so we can make more supply. Less compatibility issues, we can make O- blood, which means everyone can have that blood. And non-infectious disease transmission. People in developed countries often give blood that might be infected with disease, HIV or anything else. Whereas if we make our own blood, artificial blood, we can make sure that those blood that is made, it doesn't have disease. So we don't accidentally transmit infectious disease. How far are we? We can make artificial plasma. We can also make perfluorocarbon emulsion, and that is an oxygen carrier, short-lasting oxygen carrier. It's similar to hemoglobin, but not as good. But we will soon, so maybe in the next 10 years, be able to make artificial hemoglobin. That's the better version of perfluorocarbon emulsion. But that should be there in about 10 years' time. Then we also have, we talked about this as well, the described current theories about the process responsible for the movement of materials through plants in xylem and phloem tissue. First, xylem again, we had this diagram. We have a example of how salts are they're actively pumped into the roots. So salts are pumped into roots. And remember, if you have lots of solute in one place, if you have low solute in the soil and high solute in the actual root, then when you have that, you have through osmosis, you have water traveling into the root as well. That's how water enters the root through osmosis. Once it's in the root, it's going to climb up the stem itself. And remember, there was two important parts, cohesion and adhesion. Cohesion was when they're kind of harnessed to each other. So when water molecules, which are these ones in blue here, when they're harnessed to each other, so these white lines were the harnesses between the water molecules. And then we have adhesion, which was when your water molecules were also clinging on to the actual sides of the wall as well. And that was all there to make sure that you don't have that drop happening, because they're climbing against gravity. If you didn't have adhesion and cohesion happening, you would be going down. So that doesn't happen. And the reason why is because we have cohesion and adhesion. And remember also then this whole thing goes, so the stream goes from the roots all the way to leaves. And at the leaves you have that transpiration happening. So transpiration was evaporation. But the thing is, if you have one or two molecules and transpirating, that means they're going to basically pull on the other ones, because that harness that they're attached to means if one or two of them drop, that means that harness is going to pull on all the other ones. So you're going to have the other ones being pulled up as these two transpirate or evaporate. You can imagine these water molecules jumping, and then the harness pulls the other ones along. That's why we call it the transpirational pull. So that was the name of the mechanism, transpirational pull. And the reason why is because these are transpirating, but because of cohesion, you have, once these are transpirated, once these evaporate, that pull will make sure all the other ones come up as well. Then we have how sugar travels. And sugar, remember these words, source and sink. Source was the area where sugar is produced, and sink was the area where sugar is needed. So a source might be the leaf, and sink might be the roots or the stem. First we have the sugars being actively pumped from source, so from the leaf, into the phloem. So that happens right here. What happens next is water follows through osmosis. So because now there's a high solute in the extra phloem, you're going to have water going from xylem to phloem. And you can imagine now you've got lots of water and lots of sugar in that tube. That means there's going to be lots of pressure, because there's going to be lots of stuff in a tight space. And what that's going to do is it's going to be creating a high pressure area close to the source. And a high pressure area always moves from high pressure to low pressure. And the low pressure area is at the sink itself. So it's going to move from high pressure from source to sink for that pressure flow. And that's that third part here. So sugar is moved from source to sink for that high pressure. And once it's at the actual source, it's going to unload into the sink cell. And it's going to do so actively. It's going to be pumped through over. And once it reaches the sink cell. And we call that translocation. So our sugar is translocated. It's changed its location, transport location. So it's changed its location from the source to sink. And it's done so through this mechanism I described earlier. Then we also have, you have to draw the cross-sectional area of xylem and phloem. Remember, cross-sectional, that means if you, for example, were to cut a stem, the green stem, and you look at it from the top, that's what we call cross-sectional. The things you need to be able to draw a label is just the stem itself, and then you have the pith in the middle. That was the pith. And then you would have that vesicular bundle, which was this bundle here. And in the bundle, so the bundle itself is made up of three parts. You got your phloem on the outside of that bundle. You have your xylem on the inside of that bundle. And in the middle you have your cambium tissue, which is a tissue that can divide into either phloem or xylem. So remember that, you should be able to draw that. And also the longitudinal, so this is the longitudinal of the phloem. Longitudinal is like looking at it from the sides, cutting in and then looking from the sides. So a phloem is made up of three parts: companion cells, sieve tubes, and sieve plates. So things here in brown as your companion cells. What they do, they help with active pumping of sugar. So remember I said that sugar gets pumped from source into phloem. That happens here at the companion cells. So these pump your sugar into your sieve tube. Your sieve tube was that tube which connects the source to sink. And then you have your sieve plates in the middle as well. And they have holes to make sure that sugar can pass through. So that was your longitudinal section of your phloem. So hopefully that was helpful, and we're going to do the next topic chapter, regulating substances, next. Thank you.